Hello everyone and welcome to Bruker Cellular Analysis conference. You might be wondering, what is Bruker Cellular Analysis? Although the name is new, the company has been around for over 10 years. Previously, you knew us as a Berkeley Lights and Isoplexis. Bruker Cellular Analysis is the new name of the company formed by merging Berkeley Lights and Isoplexis. And now we are part of Bruker. Regarding products, we launched our first platform, the Beacon Optofluidic system, almost 5 years ago. Since then, it has transformed how scientists develop cell lines, discover antibodies, and profile T cells. 2 years ago, we acquired Isoplexis and their IsoSpark platform to complement our Beacon platform and enable single-cell high-plex proteomics. Both the Beacon platform and IsoSpark are widely used in the scientific community. More than 400 instruments have been installed worldwide in biopharma companies, academic institutes, and CROs. Researchers have validated both platforms with over 250 peer-reviewed publications. And nothing is better than peer-reviewed publication to showcase that platform just works. For the rest of my talk, I will focus only on Beacon platform. The Beacon lets researchers get real-time data on many aspects of single-cell function. Understanding how single-cell work is important for advancing translational and clinical research. Last 30 years or so, flow cytometry has helped us characterize cell surface protein and enable immunophenotyping. Then came single-cell sequencing, and it has helped us study cell genetics and gene expression. But there has been a problem. No single tool can provide data on many functions of a cell. Call it a collective life for each cell, if you will. The Beacon platform provides a breakthrough and solve this problem. The Beacon enables us to collect data from multiple functional assays on a same cell. And as a result, help us understand the life of single cells. Conventional technologies like FACS or sequencers look at individual part of single cell function and do not provide the whole picture. Most of them enable endpoint assays, providing information on either secreted protein or cell-cell interaction or cell surface marker or gene expression. It is important to note that you need multiple platforms to get data on these functions, and data points are coming from different single cells, not from a same cell. It's a very big point, so I'll repeat it again. All this data, although you are using multiple different platforms, you're still getting this data from different single cells. Whereas when you compare it with a Beacon platform, we provide multi-modal data from the same live cells over multiple time points. It gives data on morphology, cell-cell interaction, surface protein, intracellular proteins, secreted protein, cytotoxicity, and genomic analysis, to name a few. And again, from the same live cells. And this has been made possible by two main innovations: Opto-Electro Positioning technology and NanoPen horizontal design. Our Beacon platform chip consists of phototransistors. Shining light of them changes the charges on this phototransistor and allowing you to move cells. Yes, you heard it right. You can have complete control on the movement of cells as well as on assay beads, and thereby providing you ultimate freedom to design your own experiment with the Beacon platform. As you can see in this video, we create a small electric cages, and these cages allow you to accurately place single cells at individual nanopens. Additionally, chip has a design to allow rapid fluid exchange, meaning you can change media as well as assay reagent without disturbing cells in NanoPens. This allows you to run multiple assays one after the another with same cell. And the system has inbuilt high resolution microscope, which enables real-time data for each and every assay. Here are few examples of precise control over the movement of cells and assay beads. In the video on the left, you can see how we are using inbuilt fluorescent microscope to select specific cells. Here we are selecting CD3 and CD8 double positive CAR T cells. And once they are selected, we are moving them individually to individual NanoPens. And then in the video on the top right, you can see how cytokine capture beads, T cells, and target cells are moving into NanoPen one after the another. And then in the video on the right bottom, you can see once you are done with a functional analysis, you can select a NanoPen and take all the cells out from that NanoPen to conduct a next experiment with it. The power of Beacon platform is its ability to run multiple assays on a same cell in a high throughput manner. So here is an example with T-cell profiling application. You can profile tens of thousands of T cells with 3 or more assays, including dynamic cytotoxicity, multiplex cytokine secretion, and cell surface phenotype. For dynamic cytotoxicity, using caspase-3 as a substrate, as shown here with a teal color, you can determine when T cell kills a target cell and at which rate. And by doing that, you can determine if the T cell has low or rapid cytotoxicity phenotype. You can also assay serial killing capability of T cells without getting exhausted, which is really important for cancer treatment. And then you can quantify up to 3 cytokines in one single experiment. Followed by that, you can add antibodies targeted to a cell surface marker and quantify those cell surface protein in a serial assays. And once you have done all these, you get a comprehensive understanding of T cell function. And with all these data has been generated, you can plot it into a UMAP to identify clusters with a specific cellular behavior. For example, T cells in a pink cluster highlighted here secrete moderate level of cytokine 1, high level of cytokine 2, or high level of cytokine 3, low level of cytokine 2, and low level of cell surface expression with a moderate level of cytotoxicity. Now imagine that you can extend this information to tens of thousands of cells and generate multiple cell clusters, each with a specific cellular behavior. And you don't have to stop here. The beauty is that you can even go a step further. You can select a cluster with a specific cell behavior and recover all the cells in that cluster out of this chip and place them on a 96-well plate. Then use our provided TCR-seq or single-cell 3'-mRNA-seq solution to understand why these cells behave in the manner they do. And we are the only one, or I have to say the Beacon platform is the only solution which can link functionality to the genomics for the same single cell. I'm not aware of any other solution which can provide such information. Above all, Beacon is a flexible platform and can provide end-to-end fully optimized workflow for not only T cell profiling, but also for therapeutic antibody discovery and for antibody producing cell line development, as outlined here. Let's look at them one by one. I just provided an overview of T cell profiling workflow. Same workflow can be used with the multiple stages of cell therapy development process. Cell therapy development process involve discovery, validation, and clinical translation. At the discovery phase, the goal is to find effective T cells and CAR sequences in the library. The Beacon provide a high-throughput single-cell screening method to find the best candidate quickly. At the validation phase, the aim is to confirm that cell therapies work. And Beacon provide detailed functional data to ensure the cells are potent and thereby facilitate their move into our clinical studies. At the clinical translation stage, the challenge is donor variability. Different donors can respond differently to therapies. We can analyze single cells to find a biomarker that explains why some patients respond well and others don't. Today, right after my talk, Dr. Ickmuller will discuss how Beacon facilitate their T-cell discovery work, and Dr. Zamora will show Beacon's role in translational biomarker research. So let me focus on validation phase of cell therapy. Dr. Thomas from St. Jude Children's Hospital recently published their work with Beacon on cell therapy validation phase. They discovered two TCR capable of attacking cancer-specific fusion protein, but needed further validation or further proof of functionality before proceeding toward in vivo experiments. They used Beacon to compare single-cell cytotoxicity and cytokine secretion for both TCRs. And results established that SJ14 exhibited greater level of cytotoxicity as well as polyfunctional cytokine secretion, provided the required confidence to our researchers to move ahead with SJ14 for clinical studies. Now, let's switch gears and talk about therapeutic antibody development application. Looking at challenges in an antibody discovery field, the most important one is high-value antibody targets are getting harder to find. Previously, we thought GPCR, ion channels, and membrane transporters were challenging. Now, now new targets are being explored, especially those are highly homologous, toxic, and have a low immunogenicity, making it difficult to create specific antibodies or even harder to screen them. Accordingly, characterizing antibodies is becoming more rigorous. We need to start with large variety of sequences to ensure that we find the best therapeutic candidate. In addition, costs and timelines are also increasing. To lower these, we need more thoroughly early functional screening to avoid wasting time and resources on less promising candidates. Fortunately, the Beacon antibody discovery workflow can address all these challenges. And we believe that Beacon should be the go-to method for working with difficult targets. So let's look at how Beacon antibody discovery workflow helps. First, it broadens antibody sequence diversity to increase the chance of finding a high-value target or high-value antibody. Our world-class reagents allow screening from many species, including human, mice, rabbit, as well as many B cell types, both memory B cells and plasma B cells, multiple B cell sources directly without using sequence diversity from high homologous fusions. Second, we can enable high sequencing through high screening throughput. You can automate screening of up to 60,000 single B cell in one run. And third, Beacon enables multiple customizable functional assays, delivering functionally validated lead candidate instead of just a random or a simple hit generation. You can perform binding assays for soluble recombinant targets and even a membrane-bound targets. Additionally, we are all set to launch our affinity assay by the end of this month that will relatively rank antibodies based on their binding affinity. We also provide a foundation for you to customize more complex functional assays like ligand blocking assays and internalization assays. All functional screening, once you're done with all functional screening, you can use our Opto-Seq PCR kit to recover paired heavy and light chain sequences from identified hits. Additionally, there are multiple bioinformatics solutions available to connect these sequences with all functional assay data you have generated earlier on the chip on Beacon instruments, enabling you to do a more effective lead candidate selection based on a comprehensive functional characterization. Interestingly, when you compare our Beacon antibody discovery workflow with traditional hybridoma workflow or even with B-cell sequencing first approach, both hybridoma workflow and B-cell sequencing approach takes few months for hit generation and lead selection. Whereas Beacon platform can reduce the entire process to just 2 to 3 weeks, with only one single day needed for functional screening. This fast turnaround, when combined with functional first capability and access to a broad sequence diversity, leads to a higher antibody discovery success rates with Beacon platform, and that also in a very cost-effective manner. No wonder many of our customers have replaced their hybridoma workflow with a Beacon platform. For instance, Ivona from Genovac always uses Beacon platform when working with difficult high-value targets. Now, let's move to the third application, which is cell line development workflow. So developing cell lines is crucial for bringing new therapies to patients. However, it is a long and resource-heavy process, usually take 20 weeks or even more and involve many steps. There are two main factors affecting the cost and risk at each step. First, the number of clones advanced to the next step. And second is the functional test used to evaluate and select these clones. Traditional methods carries a lot of risk into a later stage of development process because critical performance assays like quality and stability are pushed toward the later stages. However, the Beacon platform is changing this game completely by moving functional assays to the earliest stage of clonal selection. And this is possible due to two main reasons. First, our NanoPens are more than 5 logs smaller in size than wells in a multi-well plate. With high cell density, NanoPens microenvironment closely mimic that of a bioreactor and improves our limit of detection. Second, Beacon offer multiple functional assays for growth, productivity, and quality at the early stage of development. In fact, Beacon provides end-to-end optimized workflow for cell line development. At the first cloning stage itself, cells are counted for growth and checked for monoclonality. Beacon platform keeps complete electronic and visual record from before cloning through the final assay and export of clones. Our customers at Amgen have published studies showing more than 99% monoclonality achievement with the Beacon. We also provide a fluorescent spotlight reagents that bind to protein and visualize its concentration, and enable on-chip quantitative productivity measurements. Additionally, we can provide quality assay for detecting product aggregation and measuring percent heterotimer concentration for bispecific antibodies within the first 5 days of cloning. Actually, I'm super excited to hear Dr. Wang's talk later today, where he will present their work on bispecific assay development. On the top of all these, we also offer clone stability measurement within the first 4 to 6 weeks of single cell cloning. Unlike other traditional methods that do this near the end of their clonal selection process. As a result, Beacon routinely delivers 30% reduction in campaign timelines, increased production with selected clones, and lower downstream cost as compared to the traditional methods, as shown here in a data from peer-reviewed article published by Amgen. So taken together, Beacon is a very flexible platform with varying throughputs all the way from 1 chip per run per instrument to 4 chips per run per instrument. Provides complete freedom to you for assay customization, as well as provide you off-the-shelf broad menu of assays for antibody discovery, T-cell profiling, and cell line development. And all these comes with streamlined data analysis modules with our Beacon platform. And that is why Beacon platform has fast-growing scientific community. Don't miss out. We have more than 50 technical experts ready to make you successful with this cutting-edge technology. Actually, few of them are available today at our booth in this conference. So take some time out and talk to them at our booth. With that, let me pass this back to Israel, and I will look forward to joining you again during our roundtable discussion. Thanks, everyone. Have a good day.